Well, aloha everyone, and welcome back to Biology 101 here at Chaminade University. Today we'll be covering the material presented in Unit 10, or the expression and regulation of genes. We'll talk about how DNA, which is the hereditary material stored inside the cell, is used and how it is transcribed into RNA. Then we'll talk about mRNA, which is the product of transcribing DNA into RNA, and we'll talk about how that's translated into proteins. Then we'll talk about how mutations affect protein structure and function, and how gene expression is regulated. So, in order for the information in the cell to get to the point where we are actually making a functional protein, we have to go through several steps. So DNA is the molecule that contains the blueprint for all of the genes and all of the proteins that are made in your cell. And proteins are the final product, the construction workers of the cell, and proteins control everything in the cell, from the cell shape, cell function, reproduction, and the synthesis of various biomolecules. So there's an overall flow of information from DNA to RNA to protein, right? And before scientists understood that genes were comprised of DNA, they had already done a little bit of genetic manipulation to determine the effect that genes had on the phenotype, or the outward appearance, of individual cells and organisms. In the 1940s, experiments on bread mold demonstrated that one gene codes for one protein. So one gene makes one RNA molecule, makes one protein, and that's the part that's missing there, is the RNA. And proteins are the molecular workers of the cell. They're the ones that do all of the cellular functions. They form many of the cellular structures. They're biological catalysts for chemical reactions. And so we now know that information flows from DNA to RNA to protein. Okay, so I'm going to give you an example here. So if you were to say that you had a very ancient recipe book, and in that recipe book, it had recipes for absolutely every type of food that you could ever imagine. You could consider that being your DNA molecule. The DNA molecule has a ton of different recipes or genes that code for how to make different proteins, right? But we need an intermediary molecule, that's RNA. And basically what happens is, if you want to make cherry pie, right? You want to make one particular recipe. You don't need every single recipe in that recipe book. So what you do is you take a copy of the cherry pie recipe out of that extraordinarily long recipe book. You can consider that Xerox being the RNA, right? It's still not the cherry pie. It's an intermediary molecule, but it's going to be something that you can take into the kitchen and you can use to make the cherry pie without worrying about spilling something on it. Okay? So DNA is that ultimate recipe book with all of the recipes of every kind of food mankind knows, and that's all kept in the nucleus, or in the library of the cell, if you will. And protein synthesis, which is the actual creation of that cherry pie, or in the kitchen, right, that occurs in the cytoplasm on structures called ribosomes. And so they occur in two separate spots, but that recipe that's been Xeroxed, that RNA, can actually leave the nucleus and transfer over to the ribosomes in order to code for protein synthesis. So that's kind of what's going on. Basically what happens is you're going to make a copy of that DNA that's going to leave the library or the cell nucleus and go into the kitchen or the cytoplasm, right? And in order to do that, it has to leave nuclear pores. So you might remember our cell structure talk when we talked about how we had these nuclear pores that surround the nuclear membrane. And in order for the RNA to get out of those nuclear pores, it has to pass a couple of checks. And basically the gate guards at those nuclear pores check to make sure that it is RNA by looking at the structural differences between RNA and DNA. So RNA is generally single-stranded. It uses a different sugar than DNA in the nucleotide, so the sugar is actually a ribose instead of deoxyribose, so it has one extra oxygen. Also, it's going to contain the base uracil, so U, instead of thymine. So if we see thymine, we know we're working with DNA, and if we see U, you know you're working with RNA or uracil. Okay, so just a quick comparison of DNA and RNA, and this is all stuff I've already pretty much told you, but DNA is generally double-stranded. RNA is single-stranded. DNA is comprised of deoxyribose. RNA is comprised of ribose. We have the same four bases, A, T, C, and G, except in RNA we have A, U, G, and C. So U is going to take the place of T. And the binding of DNA to DNA is going to be A to T, T to A, C to G, and G to C. And you should know that because we've done some of this DNA work extensively in several other lectures. Remember, the straight lines bind. A and T both have straight lines, and C and G both have curved lines, so they're going to bind. But over here, if we're looking at RNA to DNA, so A is still going to bind to T to DNA, right? DNA still has T, but A in the DNA is going to bind to U in the RNA. right? The other molecules are going to bind the same way. And if we're doing RNA to RNA, it's going to be A to U and U to A. So anytime that you see a U on one side and a T on another, you know that you have an RNA DNA complex. All right, and so what's the function? So DNA, as I mentioned, is that cookbook. It has all of the recipes. So those are genes, right? It contains genes, and it has a sequence of the bases in most genes are going to determine the amino acid sequence of the protein. So what is happening, it is basically going to be the blueprint, and then a copy of that is going to be made in the form of messenger RNA. So that's going to carry the code for the protein from the DNA to the ribosomes. That's going to actually leave the nucleus and go into the cytoplasm where it's going to instruct protein synthesis. We also have ribosomal RNA and transfer RNA. So all of these different molecules that are involved in the creation of proteins are going to be RNA molecules. So messenger RNA is what we talk about when we say DNA, RNA, protein. But the protein is actually going to be composed by ribosomes and transfer RNA as well, because transfer RNA is going to be carrying one amino acid at a time to the ribosomes. So we have three different types of RNA that are involved in protein synthesis. Again, all of these are RNA molecules. Messenger RNA, or mRNA, that's going to be the actual Xerox of the recipe that's going to carry the genetic information to the ribosome, so that intermediary molecule. We also have transfer RNA, and there's several of these, one for each amino acid, actually one for every codon combination, but I digress. So there's several of these, and they're going to be able to bring amino acids one at a time to the ribosome and then transfer the amino acids to the growing polypeptide chain. We also have ribosomal RNA. Ribosomal RNA is structural, it's part of the ribosomes themselves. So we have the small and the large ribosomal subunit. Those themselves are comprised of ribosomal RNA. All right. So if we talked about DNA being the old recipe book, right? DNA is going to provide the instructions for the synthesis of many, many proteins, right? So DNA has a ton of genes on there, and each gene is going to code for a particular RNA intermediate, which is going to code for a particular protein, right? And RNA has many other roles besides protein synthesis, but we're just going to touch mainly on protein synthesis today. But I wanted to just demonstrate to you some of the other ways that RNA is used. So it can be used as the genetic material in viruses, like HIV. We also have enzymatic RNA, those are called ribozymes, and those actually are going to be cutting apart various molecules of RNA and splicing them back together. And we have XIST RNA, which, as you may remember, ladies, you have an extra copy of an X chromosome as compared to the males in the room. And that means that you are going to preferentially shut one down in every one of your cells so that you don't end up duplicating or coming up with duplicate amounts of protein as, say, the men. And you do that by XIST RNA. It's a specialty type of RNA that shuts down one X chromosome in all female mammal cells. We also have microRNA, which is involved in the immune response, so regulating development and also, again, immune response fighting diseases. Okay, so this is an overview of the three major types of RNA required for protein synthesis. I'm going to give you about 10 seconds just to kind of read over this slide before I jump in. All right, so at the top here we have messenger RNA. And mRNA is divided into codons. These here are sets of 3 nucleotide bases that are each going to code for one amino acid. Okay, and that's going to get squished in between a large ribosomal RNA subunit and a small ribosomal RNA subunit. So they're going to sandwich this messenger RNA. And then we also have the tRNA here, which has an anticodon region that's going to be specific. There you go, specific to, so it's going to be equal and opposite to the codon. So an anticodon would be the exact opposite of the codon, and it would also contain an amino acid. And so when the anticodon meets the codon and they match perfectly, then this amino acid is what's going to get attached to the developing amino acid chain. Right? Again, ribosomal RNA is involved in shuffling along the messenger RNA and putting all of these in spots, so that it has the tRNA binding sites here and the amino acid binding sites and a catalytic site whereby we're going to add one amino acid to the developing chain. And again, tRNA is involved in transferring the attached amino acid over to the developing chain once we have a complete match of the anticodon and the codon region on the mRNA. So anticodon tRNA, codon region on the mRNA. All right, so messenger RNA is very important, right? It's the Xerox of the recipe for cherry pie or whatever protein you're trying to make. So it carries the code for the protein synthesis. Now, mind you, it's not the protein itself, of course, but it is going to be able to create the protein by becoming the recipe, right? And so it's going to leave the nucleus, right? It's originally created from DNA. It leaves the nucleus. It goes out to the ribosomes, which are in the rough endoplasmic reticulum. There's also some that are free in the cytoplasm. But my point is that it's outside of the nucleus. And then we are going to have ribosomal RNA, which is going to sandwich that messenger RNA and then going to read the codons, which again are groups of 3 base pairs of mRNA. So 3 base pairs equal a word. And that's going to specify what amino acid is incorporated because the tRNA has the anticodon to that codon sequence that's going to bring one amino acid at a time. Again, the ribosomes have 2 subunits, one large and one small. The large has the binding and catalytic sites needed for protein synthesis. Transfer RNA is responsible for bringing the amino acids over to the ribosomes. Again, each tRNA has a specific anticodon region, and that's going to bring one unique amino acid. So tRNA carries one amino acid at a time. It always carries the same amino acid, but there's a lot of different types of tRNA with different anticodons that are going to perhaps bind to the codon sequence and bring in a different amino acid. All right, so we have 2 major processes from which we get protein from DNA. The first is that we have to copy DNA into RNA. That's called transcription. And it's called transcription because we are transcribing in the same language. We're still in the language of nucleic acids. And again, this happens in the nucleus. After that, we have the process of translation. The process of translation occurs in the cytoplasm, and we are changing the language. We are going from nucleic acids to amino acids. All right, so this is the overview of the information flow. Here's the genes. Right, this is a long strand of DNA in a chromosome, right? There's going to be plenty of genes on there. So lots of recipes in this recipe book. We only want this recipe. We're going to make a Xerox of it, shown in red, right? So the Xerox of the gene is the RNA. And the way that the RNA is made is by the sequence of the DNA. So the RNA makes a complementary sequence to the DNA that is being transcribed. Once it's transcribed, then it is checked to make sure that it is messenger RNA by these gate guards at the nuclear pore. Then it leaves the nucleus, where it's going to drive translation by having a ribosome, actually multiple ribosomes, are going to attach. And that's going to, so this is just one, but as it comes out, we'll have several. And as they slide along, they move one tRNA into the anticodon-codon pairing over and over and over, adds one amino acid at a time to this developing protein. Okay, so the flow goes basically DNA, RNA to protein. Again, 2 major processes. The first is transcription, where we are taking DNA and making RNA, and we're going to use an enzyme called RNA pol or RNA polymerase for that. Basically, it makes an RNA polymer, so that makes sense, right? And for this, we're going to be using the DNA as a template. The RNA is going to bind to the DNA, and the RNA molecule is going to be synthesized from the DNA, again, using that as a template. Then that RNA leaves, goes out to the cytoplasm where the process of translation occurs. Translation is going to start with mRNA and create a protein molecule, and it's going to use ribosomes. It says one, but there's going to be several attached generally. And they also is going to require tRNA. Remember, each tRNA has an anticodon that matches a codon, and if it happens to fit perfectly, then that is the amino acid that is going to get added. All right? This whole thing is called the genetic code. So the genetic code uses 3 base pairs to specify an amino acid, right? And so those are codons. And the genetic code translates the sequence of bases and nucleic acids into the sequence of amino acids in proteins. All right, so how does that work? So again, DNA and RNA have 4 different bases. They have A, T, C, and G for DNA, A, U, C, and G for RNA. And we have 20 amino acids, but we have only 4 bases, right? So statistically, the smallest number of bases that could combine to yield a different sequence for each of the 20 is 3, because a 2 base pair only produces 16. But that's interesting because a 3 base pair creates 64 combinations, and we only have 20 amino acids. So that allows for a little bit of overlap in this system. What that means is that there are going to be a couple of different anticodons that are going to code for the same amino acid. That allows for a little bit of wobble. Again, the bases in the mRNA are read in triplets. Those are called codons. And the way that these were determined of which codon makes what is they would compose an mRNA strand entirely of one nucleotide, and then entirely of another nucleotide, and then entirely of a repeat of sets of 3, whatever set of 3 they put together. And then they would get a protein that was entirely comprised of whatever amino acid. So if we do all U's, then we're going to get phenylalanine. So that concludes the triplet UUU gives you the code for phenylalanine. But as they put in every set of repeating triplet, then they could one at a time deduce which of those codes for which amino acid. All right, so each mRNA molecule has a start codon and then multiple, well, at least one, but there are three different options of stop codons. So you have to start somewhere and you have to stop somewhere. So basically what happens is that we hit a promoter region and then a whole thing assembles, and then we go down to the start codon where AUG is going to code for methionine. So every single protein is going to start with methionine as the amino acid, and then from there we're going to shift over one codon and add one amino acid at a time until we reach a stop codon, which does not code for any amino acids. It simply breaks apart the chain. Right? And as I mentioned, each codon specifies only one amino acid, but multiple amino acids can be specified by different codons. So some amino acids are specified by as many as 6 different codons because of the fact that we can make 64 combinations but only have 20 amino acids. Okay, so this is a codon dictionary. For the purposes of this class, I will not expect you to read one, but I do want you to understand one. So basically, if you were looking at, for example, previously we were just looking at UUU, which is phenylalanine. So the first base pair is found here. First base pair is here is U. Then we're going to do the second base pair across the top, and then the third base pair along the side. So this would be U, U, U. Right here is phenylalanine. But if we wanted to look for the start codon, for example, that's AUG. So let's go down to A. Okay, stop here. Go across again to U. All right, we stop here, and then down to G. That's here, AUG. So if you wanted to do, I don't know, CGG, right? We go to C, then we go over to G, right? So here's CG, and there's 4 of them, and we go down to CGG. That codes for arginine. So you're able to, given the codon sequence of an mRNA molecule, determine the protein sequence that it will code for, right? And again, we have 3 different stop codons, UAA, UAG, and UGA. All right, so how do we decode what's going on with the codons and which amino acid gets put where, right? And that's basically the job of tRNA and ribosomes. So mRNA is just going to be the code itself. It's just the recipe, right? If you mess up making the dinner, that's not on you if you didn't follow the recipe. So the mRNA is the recipe, and the tRNA and the ribosomes are the cooks, if you will. And each tRNA molecule has 3 exposed bases. Again, that's an anticodon, which is going to be exactly opposite or complementary to the codon base in the mRNA. And if it fits, then it's going to be the one that's going to be chosen to add that amino acid. So transcription is where we're going to start. So transcription is the process of creating RNA from DNA. Again, we are transcribing. That means we are using the same language. In this case, it's nucleic acids. And it has 3 stages. The first stage is initiation, and then elongation, and then termination. Right? And so for initiation, we have to have a promoter region. A promoter region is a region upstream that's going to mark where transcription is to be initiated. This is the start codon. This is where you would scroll down the recipe. The first part would just be all ingredients. Then it's step 1, right? This is where you start. And then you have the body of the gene, and that's where the entire elongation of the RNA strand is going to occur. And then at the end of the gene, you have a termination sequence. And at that termination signal, that's when the mRNA is going to fall apart from the ribosomes and the whole thing is going to terminate, and the protein is going to be, the RNA is going to be released. I apologize. I said mRNA here, and mRNA is being created at this point, and there are no ribosomes. So I apologize. I just kind of switched a little bit. We're talking about the process of transcription here. And that's going to begin when the RNA polymerase is going to bind to that promoter region that I just talked about. That promoter region, again, is upstream of the gene, and it has a site that's going to bind RNA polymerase and has multiple different, what we call transcription factor binding sites. And anything that's going to assist in transcription is called a transcription factor. And these are some sort of response elements that are going to be helpful in increasing or decreasing the rate of transcription of particular genes. So once RNA polymerase is bound to the promoter and the DNA molecule is unwound, the DNA is opened so the RNA pol can go ahead and create that RNA molecule, then the strands are separated out at the beginning of the gene sequence. So this is an overview of the synthesis of RNA from DNA, which is known as transcription. So first we have initiation. The initiation occurs right here at the beginning. So there's going to be a little region upstream where the RNA polymerase is going to bind to the promoter region, and that's going to separate out the double helix right near that promoter region. So this is RNA pol. RNA polymerase is going to be moving along the DNA template strand, creating the RNA from the exact equal and opposite, right? So we're making matches based on the template strand. That's called elongation. And then we're going to reach a termination sequence. Once we reach that termination sequence, then the RNA polymerase is going to fall off and detach from the RNA and release the RNA molecule. Sorry, detach from the DNA and release the RNA molecule. At the end of transcription, DNA completely rewinds back into the original double helix, so it remains unchanged, and the RNA molecule, which is single-stranded, is free to move out into the cytoplasm where translation is going to take place. Okay, so The RNA polymerase is responsible for synthesizing the RNA nucleotides one at a time by using the DNA strands as a template. And it's going to travel down. This is 5' to 3' is the direction. I'm sorry, 3' to 5' is the direction, but you won't need to know that for the purposes of this exam. And basically what we're using is complementary base pair. So as we bring in one new base to the RNA, it's going to be complementary to the template strand of the DNA. We're going to start at the initiation strand, and we're going to form RNA all the way until we hit that stop sequence. And as that happens, the RNA molecule is going to kind of drift away from the DNA templates until we get to the point where we end up with the termination sequence. And as the RNA strand moves away from the DNA strands, the helix is going to reform right behind RNA polymerase. Eventually, RNA polymerase reaches that stop sequence, right, a termination sequence. And that releases the completed RNA strand, and the whole process means that it detaches from the RNA, I'm sorry, from the DNA rewinds, and the RNA is now free to leave and go into the cytoplasm, right? The RNA polymerase is going to get recycled, so it's going to bind over and over to another promoter region of another gene, or maybe to the same gene, and synthesize another RNA molecule. Okay, and so this is what this looks like. So here's the gene, right? Here's the DNA strand, as shown in blue. And as we are in this direction of transcription, you can see that because they're getting longer. Here's our termination sequence, or the end of the gene. And you can see that we have multiple different strands of the RNA molecules that are growing at the same time off of the same gene, right? And then these are going to then leave and end up undergoing translation, or the process of creating proteins. Okay, so messenger RNA synthesis is drastically different between prokaryotes and eukaryotes. So everything that I've been talking about when I say leave the nucleus and go out into the cytoplasm, that's all eukaryotes, right? Because prokaryotic cells don't even have nucleus. And so prokaryotic genes are a lot more compact, and they're generally all adjacent to one another. So genes for related functions are going to be A, B, C, D, all in a line. That is not necessarily the case for eukaryotic cells. Genes that are all related to the same function could be scattered all over the genome and regulated by completely different promoter sequences. All right, so prokaryotes, again, they have no nuclear membrane, they have no nucleus at all, and so translation and transcription happen at the same time. So sometimes an mRNA molecule is separating from the DNA during the process of transcription, and at the same time, ribosomes are going to jump on it immediately and translate that mRNA into protein. So there's no separation in time or space between the creation of the mRNA molecule and the creation of the protein. However, in eukaryotic cells, it's drastically different. DNA is contained in the nucleus, and the creation of mRNA happens in the nucleus. Ribosomes, which are responsible for protein synthesis, reside in the cytoplasm, so they're in a totally different location within the cell. And also, genes aren't really clustered necessarily. They can be dispersed all over the chromosome. So just because or across several chromosomes. So just because they're involved in similar functions or in the same metabolic pathway doesn't necessarily mean that they're going to be found underneath the same promoter or even on the same chromosome. And mRNA molecule that's formed is going to need a little bit of processing to be translated into protein. So it's not going to be immediately ready as the prokaryotes are. All right, so this is what a prokaryotic chromosome looks like. So here we have the DNA molecule. From the DNA, we're making RNA. From that mRNA, we're making protein right there at the same time. Right, so again, RNA polymerase is opening up the DNA. The RNA is being created from the DNA template strand. Ribosomes are attaching and creating protein all at the same time. So that's simultaneous transcription and translation. Again, that only happens in prokaryotes. That does not happen in eukaryotes. In eukaryotes, we have an additional step that has to happen to the mRNA. So we have to have mRNA processing. We have regions inside the mRNA that are going to be translated. Those are called exons. And regions that are interrupted, basically. These are called introns, or interruptions. And you can picture this like video recording an entire TV episode, but you want to eliminate the commercials before you watch it. So you're going to cut out the interrupting factors, and then you can watch it commercial free, right? And so the exons are going to be the TV series, and the introns are going to be the commercials. All right, so the long strand of mRNA that's made first is called precursor mRNA. The precursor mRNA is going to have a little bit of processing. First, we are going to have some nucleotides added at the beginning and at the end of the mRNA molecule. That's going to form a cap and what we call a poly-A tail, and basically that's going to form a ring. So now that mRNA molecule is going to be a ring because the cap and the tail are going to bind to one another. That's going to assist in the longevity of the mRNA molecule because it's going to be degraded pretty much as soon as enzymes are able to start getting away at the tail. And so that's why I put additional poly-A at the end of the tail so that those enzymes that are degrading it are going to have something to eat while we're still processing and translating that mRNA molecule. All right, so we have specialty enzymes in the nucleus that cut out introns and splice together exons. That makes the mRNA that's going to leave the nucleus. So intron-exon splicing happens in the nucleus, and then the mRNA molecule, which is finalized, so it's already been spliced, is going to exit the nucleus through a pore. And that pore has gate guards, right, that says, are you RNA? Are you sure? Because DNA is not allowed out. And then it's going to find a ribosome in the cytoplasm, either associated with the rough endoplasmic reticulum or free in the solution. All right, so this is what intron exon splicing looks like. And just to let you know, this is a very simplified version. So we're going to remove 2 introns here. So we have 3 exons and 2 introns. And when we remove those 2 introns, we end up with a significantly shorter sequence. We've also added a cap and a poly A tail here. And of note, you could preferentially splice out this intron and not that one, or this one and not that one, and end up with alternate isoforms of this mRNA molecule. So for example, if you picture that you left in commercial 1 but cut out commercials 2, 3, and 4, you would have a totally different show than if you left in commercial 2 and cut out 1, 3, and 4. Or if you cut out all of them, right? You'd have different lengths of time, and things would move differently, even if it was the same length of time, because you would have a commercial interruption at a different point in time. So if you were to play them simultaneously, you would be seeing different things on the TV. All right, so what's the point of having this intron exon gene structure? Well, one of the main reasons is what I just described to you. It's called alternative splicing, the ability of a cell to make multiple different protein variants from the same gene. And so this is an exception to what we talked about previously. We talked about one gene, one RNA, one protein. In this case, you have one gene, multiple very similar, but slightly different because of alternative splicing events, proteins. Another function of this is that the fragmented genes might provide a way for evolution to occur. So it might be an influence in evolution. So if we end up with breaks in the chromosomes that occur in the introns, exons might remain intact and spliced into other chromosomes, for example, and that might allow us to create new useful proteins that are currently not created. So that's one of the purposes of intron-exon splicing. All right, so we are moving on now to translation. So we just talked about transcription. Again, that's when we were making transcription is making RNA from DNA. We're moving on to translation. Translation is using mRNA to make protein. But just like transcription, translation has 3 major steps. Again, initiation, elongation, and termination. And just like transcription, you need to have a complex form at the beginning in order to induce translation. So it's called a pre-initiation complex, and there's multiple different things that have to happen, but basically we have a UAC anticodon, which is going to be on the methionine tRNA, and on the actual mRNA molecule we have an AUG start codon. These two are going to have to bind together. Once they bind together, then we're going to have the small and the large ribosomal subunits binding, and then we're going to be able to initiate translation. So the large ribosomal subunit is going to attach the small subunit with the mRNA pinched between. You can picture this kind of like a zipper on either side of the metal chain, right? And that's going to be holding the mRNA again, and the tRNA is binding to the mRNA, the anticodon-codon interface. And that's going to allow the very first amino acid to be added. In this case, that'll be methionine. In all cases, that'll be methionine. So once the ribosome is fully assembled and ready to begin transcription, so that looks like this. So everything we just talked about was getting here. First, we have the mRNA and the tRNA are all going to bind. So first we have the small ribosomal subunit and the tRNA, then the mRNA comes in and binds, then the large subunit sits on top, and now we are all ready to start. Okay, then we'll enter into elongation. Elongation is when amino acids are added one at a time to the growing protein chain. So we have the next tRNA molecule, and multiple different tRNA molecules are going to bind, but are going to try to bind, but only one is going to fit like Cinderella's slipper, right? And so once the tRNA with the correct anticodon base pairs is going to pair with the second codon in the mRNA, then it's going to be bound to that second site. And then the catalytic site, which is at that top unit, that's, hold on, let me go here. Okay, I'll go backwards then. The catalytic site is up here. Okay, so methionine would be the first amino acid. If the second tRNA to bind has valine, then that valine is going to be the acceptor of methionine. So methionine is going to get added to the valine, and now this one is free and can be discarded to go find another amino acid. And at that point, this tRNA, which is bound to the peptide chain, will shift over to the empty slot, and that will allow for another tRNA to bind over and over and over. So when this tRNA does find its binding to the codon and anticodon interface, then this histone is going to become the acceptor for valine and methionine. And then eventually we're going to hit a stop codon, and when we do, the whole thing is going to disassemble and release that protein into solution. Okay, so let me go back to where we were. So basically what happens, we were just talking about the catalytic site and how the catalytic site is going to allow the transfer of methionine to the new amino acid. It breaks the bond between methionine and its tRNA and forms a peptide bond between that amino acid and the next amino acid. And then the whole thing is going to shift over. So the empty tRNA is released and the ribosome moves down one mRNA codon. The tRNA also shifts down into the next site. And now the first binding site is open, awaiting that tRNA. So now a third tRNA with the same anticodon binding to the third codon of the mRNA is going to enter. So multiple tRNAs are going to enter and try to bind, but only the one that has the correct sequence is going to bind, and then it's going to accept the growing polypeptide chain, and that process is going to be repeated over and over until we reach our stop codon. Once we reach the stop codon, there's several factors, these are releasing factors, that are going to release the completed polypeptide chain and also the mRNA and going to disassemble the large and the small subunits. And no tRNA binds to the stop codon. That means no amino acid is coded for by the stop codon. All right, so each gene in the DNA codes for a single protein or a single family of proteins if you talk about protein variants. And transcription is going to produce the mRNA strand using DNA as the template strand. Then the mRNA strand leaves the nucleus through the nuclear pores, first it undergoes processing, and then it leaves the nucleus, and then it associates with the ribosomes in the cytoplasm. And then transfer RNAs from the cytoplasm they've already been previously loaded up with their appropriate amino acids, and then they're going to bind to the anticodon sequence. And eventually, they're going to be accepting the polypeptide chain one at a time and then releasing it one at a time until eventually we get reaching the stop codon, which is going to define the site where protein synthesis is going to be terminated. All right, so let's talk about complementary base pairing because complementary base pairing is how all of this works. So when DNA is bound to DNA, we have A to T, T to A, G to C, C to G. You may remember previously that I said that all the straight letters bind and all the curved letters bind. But I also said that they bind because of hydrophobic interactions and their sizes. So every set of bound bases is going to include one large and one small. Two larges would be too big and two smalls would be too small. They would never meet, right? So two larges would overlap and two smalls would not meet. We also have this different number of binding of your hydrophobic interactions. So we have 2 hydrogen bonds here and 3 hydrogen bonds between G and C. And that's how G cannot bind to T and why A cannot bind to C, because this one has 2 and this one has 3. This one has 3 and that one has 2. And so by size and by charge, we're able to have binding of these base pairs through hydrophobic interactions all along here. And the fact that hydrophobic interactions are relatively weak individually, but as a whole very strong, are going to allow, similar to like Velcro, are going to allow for the DNA to wind and unwind as necessary without any changes to the DNA or damage to the DNA. So using the DNA as a template, you create the mRNA strand. The mRNA strand has these particular codons, AUG, GGA, GUU. Now, if you look upwards, these are the same as the original. So this is the strand that was used as the template. This is the complementary. These are the same as the complementary strand, except for we have U instead of T, right? So U is going to be very similar to T, but is going to be different. Now, these codons are going to bind specifically with the anticodons on the tRNA. And that's going to code for 1 amino acid. So every 3 nucleic acids codes for 1 amino acid with the exception of the stop codon. So with the stop codon, we don't code for any amino acid and we just release the developing polypeptide chain. So what happens if we end up with mutations? What if we make a mistake? That seems like a lot of pressure to make sure that every single step we get the right pairing, right? Mutations do occur. Mutations happen when we have changes in the base sequence of DNA, and they can be caused by multiple things. They can be caused by mistake strain replication or by environmental stressors. And we have several different types of mutations, but they fall into 5 major categories. The first is an inversion, and that's when the chromosome breaks at one side and the other, and it gets put back in the same location, but it gets put in inverted. We can also have a translocation, when a piece of one chromosome breaks off and then gets picked up by another chromosome. We can have a deletion, where a part of the chromosome is simply removed, an insertion, where we are adding nucleotides, or substitutions, where we have changed one nucleotide for another. So inversions and translocations are pretty major, but they don't result in the loss of any DNA. So an inversion and a translocation generally don't tend to affect the individual who is carrying the issue, but it does tend to affect their offspring, so they tend to have a hard time reproducing. And you could picture that if something that is on chromosome 5 normally now ends up on chromosome 7, that you might end up with errors in meiosis where you end up with duplicate copies of information on 5, or not enough copies of information that are carried on that chromosome because it's actually on chromosome 7 and that ended up in a different location. And so this can end up with serious issues with reproduction. And so oftentimes people don't even know that they have inversions or translocations in their genome until they go in for fertility treatment. So usually the mutations are benign if the entire gene is moved. However, sometimes a gene is split in two, and if it's split in half during a translocation, then you have half on one side, half on the other. It's not going to code for a complete functional protein. At best, it codes for two partially functional proteins, but generally it's going to code for nothing or nonsense. And an example of this is hemophilia. So hemophilia is often caused by an inversion in the gene that encodes for a blood clotting protein, and because it's cut right in the middle, it's unable to create a functional protein. All right, so deletions and insertions. So a deletion mutation occurs when one or more nucleotides are removed from the gene sequence, and an insertion is the opposite, when one or more are added to the gene sequence. And these are fairly well tolerated if they are inserted or deleted in sets of 3, meaning if we've inserted or deleted a full codon or a full series of codons, then we're going to be eliminating 1 or 2 amino acids or adding 1 or 2 amino acids. But if we have a deletion or an insertion mutation that's, say, a set of 3, or sorry, not a set of 3, say a set of 4 or 5, then you can end up with what's called a frame shift mutation, which means that everything downstream is going to be nonsense. So depending on the number of nucleotides involved, deletions and insertions can be fairly benign, or they can cause a complete misreading of the gene's codons during transcription. So if you were, or replication. So if you were to say the dog saw the cat, those are 3-letter words that are represented as these codons, right? The is codon 1, dog codon 2. If we deleted E, then it becomes the dog, wow, a cat, right? Nonsense. So it becomes separate and totally different amino acids downstream, which hopefully are benign, but might even have deleterious or harmful functions. So at best, you'll end up with a truncated protein. At worst, you'll end up with a protein that has an aberrant function. So proteins that result from deletions and insertions generally have a very different amino acid sequence than the original protein, and generally are non-functional. Almost always they're non-functional. Usually we have stop codons that are in the genome that will be triggered a little bit early, and so oftentimes a deletion or an insertion mutation that is going to change the downstream is going to then, so a frame shift mutation is going to also trigger a premature stop codon. But as I mentioned, deletions and insertions of 3 are not going to cause a frame shift, and so they might just add or subtract an amino acid, so it's going to be much better tolerated than a frame shift mutation. We can also have substitutions. So if a nucleotide substitution occurs, it's also called a point mutation. We're basically exchanging one incorrect nucleotide or exchanging the correct one for an incorrect one. And it's just going to be at one location. And that one nucleotide may or may not change the codon. It may or may not change the amino acid sequence of a protein because, as I mentioned previously, there are multiple amino acids encoded, many amino acids that are encoded by multiple codons. And so if a mutation causes a change in the codon that's going to code for the same amino acid, then there's going to be no change at all. So some point mutations are completely benign. For example, changing CTC to CTT are both going to code for glutamic acid. So you're going to get the same protein at the end, even though you have a point mutation in the gene itself. Right? So generally substitutions protein functions can be entirely unchanged. However, depending on which protein is substituted for another, right, if they're similar, it might have a similar function. If they're completely dissimilar, we might have different functions. For example, beta globin, which is the example that we'll be using, I'll show you in a little bit, multiple different mutations in this gene. But if we change it too much, we can change its structure entirely. So the amino acids on the outside of the protein have to be hydrophilic. That means they have to be able to be dissolved in the cytoplasm of red blood cells. But which hydrophilic amino acid is on the outside doesn't matter. So if we have a substitution of hydrophilic for hydrophilic, then that's fine. The resulting protein is going to function just fine. So those are called neutral mutations. But if we change it to hydrophobic, when it is supposed to be hydrophilic, this is a really big deal. So now it's supposed to interact with the cytoplasm, and it doesn't. It wants to interact with the membrane. And that's going to lead to clumping of the hemoglobin and distortion of the red blood cells that is known as sickle cell anemia. Point mutation can also introduce a premature stop codon. As I mentioned, a lot of times in the genome, there's regions that are, if you have a frame shift one direction or the other, you can get a stop codon. So there's a lot of places where stop codons are just right underneath the surface. And so a point mutation could also change it such that we get a premature stop codon, which is going to lead to a truncated or incomplete protein. There is actually a mutation in beta globin that it prevents the production of a functional beta globin protein. And that's called individuals with this mutation called beta thalassemia is the name of the mutation. They only have alpha globin instead of beta globin, and they constantly need blood transfusions in order to remain alive. So these are different mutations in the hemoglobin gene. And some of them, again, are completely normal with no disease states. Some of them cause very severe disease states and different types of disease states as well. So for example, if our original DNA template reads CTC, and a mutation arises, a mutation arises that reads CTT, well now instead of GAG, we have GAA. However, GAG and GAA both code for the amino acid glutamic acid, so we have neutral change, normal protein function. The second mutation, instead of having CTC, we have GTC. That codes for glutamine instead of glutamic acid. It is a different amino acid, but they are both hydrophilic, so it's going to be a neutral functional effect. But CAC instead of CTC allows us to make GUG in the mRNA, which codes for valine. And valine is hydrophobic, so it's a complete change of the way that that protein is going to function. So it's going to lose its solubility in water and cause sickle cell anemia. And here's another example of a mutation in codon 17, which is normally TTC. If it becomes ATC, then it codes for UAG. UAG is a stop codon. So instead of having a hydrophilic amino acid lysine, we end up with a stop codon. So we have a premature translation termination. So it's going to stop after amino acid 16, which synthesizes only a very small part of the protein, and it completely destroys protein function. And that's called beta thalassemia. Again, these individuals are going to constantly need blood transfusions because they don't have any beta hemoglobin. All right, so how is this all kind of taken into effect? So we have a lot of different genes in the human genome, 20,000 to 25,000 genes. And any given cell is going to transcribe only a small number of them. Right, if you picture that an eye cell might need different genes than, say, a heart cell, might need different genes than, say, a skin cell. So some genes are going to be expressed in all cells. For example, all cells need structural proteins. So something like beta tubulin is going to be expressed in all cells. Or genes that code for tRNAs, right? All cells require the ability to make proteins. And so tRNAs are vital in protein synthesis. And so all cells are going to have the genes for coding for tRNA turned on at all times. But other genes are expressed only in certain cells, or only at certain times in an organism's life, or only under specific environmental conditions. Remember we talked about the Siamese cat? So regulation of gene expression can occur at multiple different levels. It can occur at the level of transcription, translation, or protein activity. All right, so in prokaryotes, we're going to get this a little bit more simplified so that we can't overwhelm you too much. So we're going to talk about prokaryotes for a little bit here. Prokaryotes are limited in their way of limiting gene expression is regulated primarily at the level of transcription. So prokaryotic genes, as I mentioned previously, they generally tend to be all in a row if they are forming the same metabolic pathway. And in this way we call them operons. So an operon is going to contain multiple parts, 4 functionally related genes, right? A regulatory gene, a promoter gene, a promoter region, an operator region, and then structural genes. So the regulatory gene controls the timing and rate of transcription of all of the other genes, so it's the one that regulates what's going on downstream. A promoter is a site where RNA polymerase recognizes as the start site of transcription, so where we're going to begin the copying into RNA, and then an operator, which is going to allow access of RNA pol to the promoter, or not, right? So an operator can deny access as well. Then we also have structural genes that are going to code for the enzymes or other proteins in the metabolic pathway. So these are going to be the products, basically. So in bacteria, E. coli, you might be familiar with this. It's been a pretty nasty little bugger, and it makes the news every once in a while in food poisoning cases. Most E. coli are fine, but there are some E. coli, H57, for example, that can be very deadly. But we're just talking about a regular intestinal bacterium, E. coli, so a normal E. coli in your intestines. And this E. coli needs specific enzymes to metabolize the food that comes along. All right, so in newborn mammals, the E. coli in their intestines are bathed in milk, right? And that milk contains the milk sugar lactose. And so let's talk about the lactose operon. So lactose operon has 3 genes. Each are coding for an enzyme that aids in lactose metabolism. If we don't have lactose, the operon is shut off or repressed, and that's by a protein called a repressor protein. And we have a regulatory gene of the lactose operon, which regulates the synthesis of this protein. And when this protein is created, it represses the operon by binding to the operator sites. So, under these conditions where we are expressing the repressor is bound to the operator, so therefore RNA polymerase cannot get past the repressor. So it can bind to the promoter, but it gets stuck by the repressor protein, and therefore they're not creating lactose metabolizing enzymes. But lactose, when it's present, binds that repressor and changes its shape to open it up such that RNA polymerase is able to go through. So when lactose binds to the repressor's shape doesn't bind to the operator, and therefore the operator is open. So RNA polymerase is able to reach the promoter region and begin transcription of the genes needed for lactose metabolism. But when lactose is gone, the repressor resumes its inhibitory conformation and binds the operator, blocking transcription again. Therefore, they're not making proteins that they don't need, right? And so what does that look like? Here's our repressor, here's our promoter, and here's our operator. So when RNA polymerase is bound to the promoter region, but we have a repressor protein bound, right? So we have no lactose, so lactose is not here to pull this repressor protein away. The RNA polymerase can bind, but it can't move, so we can't get transcription of gene 1, 2, and 3. But when lactose is present, lactose itself binds to the repressor proteins. You can see this conformational shape change, so they're no longer able to bind around the operator. That allows RNA polymerase to go right on through the operator and transcribe genes 1, 2, and 3, and that allows us to synthesize the lactose metabolizing enzymes that we need to break down lactose to be able to digest it. All right, so that's very, very simplified, and that's prokaryotes. But in eukaryotes, gene expression is regulated at multiple levels. So we have multiple different layers of complexity as well in eukaryotic genes that's going to make it a little bit more complex overall. For example, DNA is found in a nucleus that's membrane bound, and we have a bunch of different types of cells in multicellular eukaryotes that might need different things transcribed at higher or lower levels. And the genome of eukaryotes is organized differently. As you may recall, prokaryotes have a circular DNA, and we have multiple sets of chromosomes, which are shaped like X's or little L's. And also our RNA transcripts undergo mRNA splicing and complex processing that prokaryotes don't undergo. So there's a lot of steps that allow us to have more fine-tuning of expression and regulation of our genes. So expression of the genetic information in a eukaryotic cell has multi-stages, multiple steps. So the first step would be transcription of the DNA, and the last step would be having the protein that performs a particular function. At any step along the way, we can increase or decrease the regulation of this protein. So cells can control the frequency at which an individual gene is transcribed, so you can create more or less RNA. And that same gene can be used to make multiple mRNAs and protein products. So we can have what's called differential splicing, or what I just mentioned previously with alternative splicing of your proteins. So you end up with different proteins from the same mRNA, depending on, different proteins from the same original mRNA sequence, which is alternatively spliced ultimately into 2 separate sets of mRNAs, or multiple sets of mRNAs. And those are going to be according to what the cell needs at that time. We can also control the translation of messenger RNAs, and we can control the stability of messenger RNAs. So messenger RNAs have a half-life. As I mentioned, that poly A tail. And the longer the poly A tail, the longer that mRNA is stable. The shorter the poly A tail, the sooner that mRNA is going to get degraded. So some mRNAs are long-lasting and translated many, many times, and others are translated only a couple of times before being degraded. Additionally, we have regulatory RNA molecules that may target mRNAs for destruction and also block translation of some mRNAs. So even though we have the mRNA transcript around, that doesn't necessarily mean it's going to get translated, because we have regulation at that level as well. We can also modify the protein. So we can start with a nascent protein that needs to be modified in order to become functional. So we'll start off with an inactive enzyme, and then we have to cut it open or excise a segment of it to expose the active site so the enzyme becomes active. Or we can phosphorylate or dephosphorylate the protein to increase or decrease the enzyme's activity. So there's multiple different ways that we can control the protein function at the protein level as well. And additionally, we can control the rate at which the proteins are degraded. So not only can we turn the proteins on and off, But we can target them for destruction as well if we have too many, or we can keep them around for longer if we have too few. So we can prevent or promote the degradation of the protein and therefore adjust the amount of particular protein inside the cell. So this is what I'm saying. We have multiple different steps by which we can control the amount of protein that is active in a cell at any given moment. So we can control it at a transcription level. So we can control how much DNA is turned into mRNA. Then we can process the mRNA differently. So if we process it such that we have alternative forms, then certain mRNA molecules with different forms will have higher levels in the nucleus or in the cytoplasm than others, right? Then that mRNA is going to leave, and that's going to, here's the cytosol, and it's going to be translated. We can regulate the stability of the mRNA, which is going to regulate the rate of translation of the mRNA, and we can create an inactive protein that has to be modified to become active. So we have another set of checks and balances just to make sure that we need this protein, then we'll turn it on or cleave it to be able to expose the active site. And when we have an active protein, which in this case is an enzyme that's turning a substrate into a product over and over and over and over, it's going to do this repeatedly. And so we can modify the protein activity of an active protein by degrading it. So we can target it for degradation, removing it from solution, which means that we will slow down the rate at which the substrate is turned into that product. So we have multiple different levels in a eukaryotic system where we can regulate the product, basically, of transcription and translation. Okay, so we can have regulatory proteins binding to a gene's promoter inside the nucleus, altering its rate of transcription. And we can have transcriptional regulation at 3 levels. We can have just the gene, the region, or the entire chromosome. So if we go back to the idea that this chromosome is going to be a cookbook with hundreds of recipes in it, right? But you want to edit it before you decide which ones get passed on. So you can edit it by removing particular recipes are not accessible, or you can say chapters are not accessible, or the entire book is not accessible. You can say, oh, we can't make desserts today, or we can't make anything from this chromosome today. So we can regulate transcription at the genetic level of both the individual gene, or at the large chromosomal segment, or at the entire chromosomal level. All right, so we have these transcription factor binding sites that are located near or on the gene's promoter region, and those are called response elements, and these are going to help regulate the rate of transcription, right? So the presence of transcription factors can increase or decrease the rate of transcription of particular genes. We can also have free radical interruption. So free radicals can create a transcription factor that binds to the free radical response element in the promoter of a gene that degrades free radicals. So if we have too many free radicals in solution, free radicals themselves trigger a response, then we're going to create more of the protein that's responsible for degrading the free radicals. Does that make sense? So a lot of these regulations happen in response to increase or decrease of particular substrates or products. All right, so another way that we can regulate gene expression is we can bind to the gene's promoter to alter the rate of transcription. So for example, in birds, if we have to increase egg production, the ovaries are going to release estrogen. Estrogen binds to a transcription factor called an estrogen receptor. That estrogen receptor binds to an estrogen response element in the albumin gene. And that makes it easier for RNA polymerase to bind and initiate transcription of mRNA, which then is translated into large amounts of albumin. And you may recognize albumin as one of the major proteins, both in our blood sera, but also in eggs to help nurture the developing embryo. All right, so we're also able to alter the genetic transcription and translation by something called epigenetics. So epigenetics are external events in addition to genetics. These are modifications to the DNA that occur that have nothing to do with the gene sequence. So we can modify the DNA, we can modify the chromosomal proteins, or we can use non-coding or regulatory RNA to change the transcription or translation levels. Now, if you picture that sometimes the recipe book is locked down and sometimes the recipe book is having Xeroxes made constantly, that would lend you the idea that the recipe book is sometimes open and sometimes closed, right? And if it's closed tightly under lock and key, then that generally is undergone by a process called methylation. So if we have a ton of methyl groups associated with the promoter or a gene, but generally of the promoter of the gene's not going to be copied into mRNA, and therefore we're not going to be able to make proteins. And so the number and location of the methyl groups on the DNA are really important in normal developmental stages, and they're also used as markers in some diseases, such as cancer. For example, things that are tightly regulated, say you're only allowed to use the part of the cookbook that makes desserts and cookies certain times, right? Only a couple days a week or something, so that you don't end up making too many cookies, right? But cancer cells, they don't care. So they open up the recipe book at any point and make as many cookies as they want. And you could consider that something like entering into the cell cycle over and over and over, which is what the cancer cells will do, right? They will override functions that will prevent them from going through multiple sets of cell division, and so therefore they end up entering into cell division many, many times, even though they already have errors in their chromosomes. So methylation is going to help shut down the book. The opposite of that is acetylation, which I don't have written here, I apologize, but acetylation would be a way of opening up the book so that we can increase the amount of Xeroxes that are made from that recipe. So increase the amount of transcription and therefore translation. We can also modify histones. And again, this is another example of epigenetics. As you may remember, DNA is wound around proteins that are histones. It's a histone octet wrapped 2.5 times, then it condenses over and over and over. And so condensed or tightly wound regions of DNA means that genes are not accessible to RNA polymerase. So it's really hard to make a Xerox of a book that is tightly closed, right? And so if the book is tightly condensed, if the DNA is tightly condensed, then you're unable to transcribe or translate it. And then we have some portions of the chromosome that are condensed that are mainly just structural and don't contain any genes at all. And so they don't need to be decondensed because we don't need Xeroxes of that part of the recipe book. It's all just, title pages, etc. But sometimes we have condensed areas that have genes that the cell might not need currently, but they might need later. So, for example, the desserts that we don't need them today, but come Christmas time, we'll be able to unlock that book. So condensed areas can decondense when those genes need to be transcribed. All right, we also have specialty RNA that's non-coding RNA that's able to alter transcription. So sometimes DNA is transcribed into non-coding RNA, which doesn't actually code for proteins. And at this point, we don't really have a strong handle on what it does, but we do know that some non-coding RNA can exert effects on gene expression. So sometimes the non-coding regions of DNA that are transcribed can actually regulate the regions that are the coding regions. And that's by something called microRNA and RNA. So mRNA, again, it's transcribed from DNA and translated into protein, and there's multiple different levels of that. How much protein is synthesized depends on several things: how much mRNA is made, how fast the mRNA is translated, how fast the mRNA is degraded, etc. And then we also have microRNA. MicroRNA are small RNA transcripts that have been cut in multiple different short strands by enzymes. And we're not really sure exactly what microRNA does. We do know that it's found in a lot of different cells and that there's variant forms in different cells within your body. And it is believed that microRNA is involved in disease defense, so in your immunology, but it is also involved in regulation of genetic transcription and translation. All right, so here's another example of what we were talking about with the X chromosome. So XIST is XIST, it's RNA, and it is involved in blocking off one of the X chromosomes in female mammals. So as I mentioned previously, females, you have 2 copies of an X. Males, you have 1 X and 1 Y if you have normal sexual differentiation and development. And what that means, ladies, is that we have double the amount of information carried on that X chromosomes as men do. And we don't want to synthesize twice as much of our products from that X chromosome. So one is going to be entirely condensed and not used at all. And what's interesting, though, is that if one of them has an error, like it codes for or doesn't code for color vision, for example, that's the one that's going to get shut down preferentially. But if there's no difference between the two, it's kind of a random chance event, and that entire cell lineage follows the same pathway. So once one cell has been turned down in terms of which X gets shut off, every cell that comes from that progenitor cell is going to have the same X turned off. Anyway, these are called Barr bodies. They were discovered by Mary Lyon, but as with many things, women don't get their say in science. And so later on, they were discovered by Murray Barr, and he got to name them. So here's a Barr body here. It's a deactivated X chromosome. We know that approximately 85% of the genes on an X chromosome that has been inactivated are not transcribed. And early on in development, one X chromosome in each of the cells is going to get shut down, and it is going to get shut down again by the regulatory molecule called XIST, which coats the entire chromosome and condenses it down into a bar body or a tight mass and prevents transcription. And again, this happens in every cell in the same lineage is going to have that same shut off. So if, for example, one set of cells in the developing embryo shuts down this X and the other set shuts down that X, from there, all of the cells from those progenitor cells are going to have the same X's shut down. And that's why we end up with like calico cats, for example, because they have clusters of cells with one X chromosome inactivated and other clusters of cells where the other chromosome is inactivated. So you have patchwork of these cells that are all descended from a single cell in the early embryo. So again, we can see this in the fur patterns of calico cats, where the X chromosome of a cat contains a gene for fur pigmentation, and sometimes you can have an X chromosome that codes for tabby cat, and sometimes you can have an X chromosome that codes for yellow, right? So if we have one fur color that's orange and one fur color that's black, depending on which X chromosome gets condensed in that progenitor cell, that's then going to give rise to everything in that entire patch of cells, then you can end up with patches of fur color on their coat, and that's what this looks like. So thank you so much for listening to me, and now you know that every time you see a calico cat, you're looking at a female cat, right? This is not something that happens in males. So I appreciate your time today. Thanks so much for listening to the end of this lecture. Aloha and happy studying.